So hello friends, today we are going to discuss the lecture 2. Okay, and in this lecture we will describe the vitamin A. Okay, so three types of vitamin. If you see the chemical name of vitamin A, then the chemical name of vitamin A is retinol. Okay, but if you see in the chemistry behind that, then three types of vitamin A derivatives have been designed. Okay, so if you see here, this is the retinol. Okay, this is the all means there will be hydroxyl group. Okay, so functional group will be the OH. Okay, if you see the second form is retinol. Okay, retinol means there will be the aldehyde. Okay, so functional group in this retinol means aldehyde group will be present. Okay, the third form is retinoic acid. Okay, so retinoic acid, oic acid indicates that there will be the carboxyl group. Okay, so retinoic carboxylic acid present. Okay, so three form of vitamins is present. Okay, the chemical name of vitamin A is retinol. Okay, but in the lab retinol also has been synthesized, retinoic acid has been also synthesized. Okay, so three form they can be converted into aldehyde as well as acid form. Okay, the next point is that if you talk about the provitamin. Okay, so provitamin, what is the meaning of provitamins? Actually, if you see here in the plant, if you see beta carotene. Okay, beta carotene. Okay, this beta carotene act as a provitamin. Okay, it is the vitamin. In this beta carotene, vitamin A is present, but it is the inactive form. Which form is it is the inactive form. Okay, but what happened here, in the presence of deoxygenase, which is the enzyme present in your body, is converted the beta carotene to vitamin A, means retinol. Okay, so it is the active form, but it is the inactive form. So inactive form is called provitamins. Okay, so it is the example. So if you see in this is the retinol. Okay, so if you so retinol, it is the retinol. Okay, so it is in the presence of reductase enzyme, it is converted to retinol. Okay, this is the OH, but it is the aldehyde. Okay, this is OL, this is AL. Okay, and if you see here, okay, so it is also converted to retinal ester. Okay, so retinal ester is in the present when comes in the lymph. Okay, lymph, in the lymph chylomicron is present. Okay, this chylomicron convert the retinal ester to retinol. Okay, and if you see here, in the blood red binding particles is present, binding protein is present. Okay, so it bind to the retinol, means groups attached from this OH, and it can deliver through blood. Okay, so it is the mechanism how retinol is converted to, means reaches to the liver. Okay, if you see again, this is the retinol. Okay, retinol means OH is present. Okay, retinol. Okay, and in the presence of reductase enzyme it is converted to retinol. Okay, retinol means CHO is present. Okay, and retinol is converted to retinol ester, and in the lymph chylomicrons convert retinol ester to retinol. Okay, and retinol is the active form, so it attached to the retinal binding proteins in the blood. Okay, so it reaches to the blood. Okay, so by this mechanism the retinal is reaches to the blood. Okay, so it is the simple mechanism behind that. Okay, so if you see here, so if you talk about the function of vitamin A. Okay, so if you see here, it only takes part in the vision. Okay, so if you see here in the retina, okay, in the retina it is present at 11 cis retinal is present. Okay, this is the chemical. Okay, retinal here indicates that it is the inactive vitamins. Okay, inactive vitamin A. Okay, and the form is 11 cis, means this type of geometry will be present in that retinal. Okay, in the presence of if you see here, in the presence of retinal dehydrogenase, it convert to 11 trans retinol. Okay, so 11 trans is the inactive form, and it is the geometry will be present in this structure. Okay, means retinal dehydrogenase convert the cis to trans here. Okay, so that's why it is the active form. Okay, this active form involved in the vision impairment. Okay, so so this is the simple mechanism, means it is the biochemistry behind that. And if you see here, this 11 cis along with 11 cis retinol, 11 cis retinol, opsin is also present. Opsin is the protein. Okay, and opsin is the protein, and if you see here, in the presence of light, sorry for that, it's the in the presence of light is converted to transform. In the presence of light. Okay, so in the presence of light it convert to, means option will be here, option will be released out, and it will convert to 11 all trans retinol. Okay, it converted to all trans retinol. Okay, so it is the simple conversion of 11 cis retinol to 11 all the trans retinol. Okay, only the chemical changes here, if you see here in this cis, means this geometry will be present. Okay, cis, either you can say cis or Z geometry, and in this is the E. Okay, so the conversion only takes part in this part. Okay, okay, so in the present, so all trans. Okay, some particles again changes one enzyme isomerase, which can convert the R trans retinol to again R cis retinol. Okay, means in the presence of light this will convert to transform, and again when light will be switched off, then this further convert to 11 cis retinol in the presence of isomerase enzyme. Okay, so it is the reverse reaction. If you see, And if you see all this in the presence of liver, in the liver, when in liver it reaches all cis retinol, there will be one enzyme name is retinol dehydrogenase. Retinol dehydrogenase, where it so it is excreted through liver. Okay, so it is the liver mechanism. It enzyme converted the 11 cis retinol to transretinol. Okay, all transretinol. And in this also, in the absence of this enzyme, isomerase work here, and it again converted to its cis form. Okay, cis form of retinol. Okay, so it is the simple mechanism. Okay, so we will erase it. And if you see again, so rhodopsin is the protein in retina. In retina there will be rhodopsin. Okay, rhodopsin convert to it is convert to lumirhodopsin, and it is easily converted to lumirhodopsin. Okay, lumirhodopsin convert to metarhodopsin first. It is converted to metarhodopsin second, and here metarhodopsin second in rhodopsin convert to all transretinol. Okay, and it is convert in the means in the presence of any reaction is will forward through any agent. So here light act as a initiator from this conversion. Okay, so it's when it's converted to R transretinal, vision will be, so there will be transduction, means transduction. Okay, so when R transretinal, when it is converted, it transduce the cells, and it releases the phosphodiesterase. Which chemical? Phosphodiesterase. So here ester bond will be, two ester bond will be present in the phosphodiesterase. Okay, this diesterase stimulated the CGMP, and CGMP activate the sodium channel in rods. Okay, and when it is converted, then when sodium channel is stimulated, they cause the hyperpolarization in the nerve cells. Okay, and this, after hyperpolarization, excitatory response to the visual cortex takes place. Excitatory response to the visual cortex takes place. Okay, and this is responsible for light. Okay, light, either it will bright as color light. Okay, so it is the simple mechanism which takes place in the rhodopsin part. Okay, so it is the starting point. This mechanism takes place by through hyperpolarization takes place, and this stimulate the excitatory response to the visual cortex. Okay, so this is the simple mechanism of vision process. Okay, so if you talk about the function of vitamin A, if you talk about the function of vitamin A. Okay, so the main function is main function vision. Main function is in proteins vision, healthy vision, for healthy vision. Okay, next is protein synthesis. Protein synthesis. Third function is prevent keratinization. Prevent keratinization. Prevent keratinization. Fourth is cell growth and differentiation. Cell differentiation. Okay, the again part is it acts also as an antioxidant. It also acts as antioxidant. The other function is German. It also thelin. Okay, so it is the simple function of vitamin A. So if you talk about the sources of vitamin A, sources of vitamin A, then if you talk about the sources, then cod liver oil. Second is shark liver oil. It is the main source of vitamin A. Okay, and third is meat, egg. Okay, egg yolk. Okay, and milk. So it is the major sources of vitamin A. Okay, carrots, papaya, mango. Okay, so it is etc. Okay, papaya and mango. So it is the sources of vitamin A. So अगर डोज की बात करोगे, मीन्स रेटिनॉल रिकमेंडेड विटामिन डोज ए की बात करोगे, मीन्स रिकमेंडेड vitamin A. Okay, then if you see here, one retinol is converted to 1 microgram. Okay, so for male it is needed in 1000 retinol. Okay, 1000 retinol means 1000 microgram. Okay, or you can also write down it. फीमेल की बात करोगे तो इन द फीमेल इट रिक्वायर्ड इन लेस अमाउंट एंड इट इज 800 माइक्रोग्राम और 800 रेटिनल. ओके, सो डिफिशियंसी की बात, सो इट इज़ द सिंपल रिकमेंडेड डोजेज ऑफ विटामिन ए. द थर्ड पॉइंट इज दैट डिफिशियंसी की बात करोगे, डिफिशियंसी ऑफ विटामिन ए. ओके, सो व्हेन you are not taking vitamin A, then symptom arises in your body. Symptoms arises in your body in which the main dysfunctioning will be the vision impairment. Okay, the second one is night blindness. So it is the form of vision impaired night blindness and corneal cirrhosis also. And the one disease is also called keratomalacia. So these are the symptoms arises when you are not taking the vitamin A. Okay, so it is all about the vitamin A. Okay, so thank you for watching this video and please like and subscribe. Okay, so thank you.